Hey guys, welcome to Fun Learning. In this video, we are going to discuss about the cell, which is nothing but the first lesson in physiology. With no further delay, let's get started. First, let us discuss about the introduction. So, under introduction, I hope so you know this sentence, which is nothing but cell is considered to be the structural and functional unit of the living body. Group of cell combined together to form a tissue. Two or more primary tissue combined together to form an organ. Two or more organ combined together to form a system. System matlab respiratory system, digestive system, all those stuffs. Now let us move on to the structure of the cell. Under this, we'll discuss about the three important things. First one is the cell membrane. Second one is the cytoplasm. And third one is nothing but the nucleus. First, let us discuss about the cell membrane. So, membrane matlab envelope. So, cell membrane is nothing but an envelope around the cell. Under this, we'll discuss about the three important things. First one is a composition, second one is a structure, and third one is nothing but a function. First, let us discuss about the composition. Cell membrane has three important components, which are nothing but protein, lipid, and carbohydrate. Among that, protein contribute almost 55% of your cell membrane, whereas lipid contribute about 45% of the cell membrane, whereas carbohydrate contribute only about 5% of your cell membrane. Now, let us discuss about the structure. So, composition, very easy. Cell membrane has three important components, protein, lipid, and carbohydrate. Now, let us move on to the structure. So, as it is a cell, we will view it under the microscope, right? So, here, when you view the cell under the electron microscope, the cell membrane has three layers. Okay, so, two electron dense layer and one electron lucent layer. Electron dense layer matlab the electron can easily penetrate to that layer. Because of that, the accumulation of electron is more in the electron dense layer, whereas in the electron lucent layer, the penetration of electron is less. So there is only less accumulation of electron in the electron lucent layer. So two electron dense layer in one electron lucent layer. So, remember this, as it is a lucent, it is L. This electron lucent layer is nothing but a layer which is made up of lipid, whereas the electron dense layer is nothing but a layer which is made up of protein. And now let us discuss about the model. So, we have three important model under this. First one is nothing but Danieli-Davidson model. Second one is a unique membrane model. And third one is nothing but Fluid mosaic model. First is Danieli-Davidson model. According to them, the cell membrane is considered to be a sandwich of lipid. That is, in between you have a lipid layer. On either side to this lipid layer, we have protein layer. So according to Danieli-Davidson model, so it is nothing but a sandwich of lipid. That is, in between you have lipid. On either side to it, we have protein. Unique membrane model. So according to them, there is and the third one is nothing but Fluid mosaic model, which is quite famous, and this model is being proposed by Singer and Nicholson. So according to them, the fluid component is being made up of lipid. Whereas mosaic is nothing but, I hope so you know this, the different type of stones which we usually seen in fish tank. So that is nothing but mosaic. So according to Singer and Nicholson, the cell membrane is nothing but the protein floats in the lipid. So according to Singer and Nicholson, the cell membrane is nothing but the protein part which flows in the lipid. Now let us discuss about the lipid, carbohydrate and protein layer of cell membrane in detail. So under lipid, we'll discuss about the two important things. First one is phospholipid and second one is nothing but cholesterol. So this diagram that shows the phospholipid. So this phospholipid has a head part and the tail part. The head part is considered to be hydrophilic in nature, whereas the tail part is considered to be hydrophobic in nature. Philic matlab it loves water, whereas phobic matlab it hates water. As a result, this head portion is towards ECF, whereas this tail portion, as it is hydrophobic in nature, it is towards ICF. And cholesterol, this square component is considered to be a cholesterol, which is generally gives a structural integrity to the lipid layer. So what is the function of this layer? So this layer is considered to be a semi-permeable in nature. That is, it allows only fat-soluble substance to pass through. Now let us discuss about the protein layer. Under this, we'll discuss about the two important protein. First one is considered to be an integral protein and second one is considered to be a peripheral protein. Integral protein are the protein which is tightly bound with the cell membrane, whereas the peripheral protein are the protein which is partially embedded in the cell membrane. So, integral protein are tightly bound to the cell membrane, whereas peripheral protein are partially embedded in the cell membrane. So, example of integral protein, in order to remember it, you have to remember the mnemonic ACE. In that, H stands for hormone receptors, A stands for antigen, C stands for cell junction protein, cell adhesion protein, carrier protein and channel protein, E stands for enzymes. Whereas peripheral protein, the example is proteins of cytoskeleton. And carbohydrate, which contribute only about 5% of cell membrane, right? So a carbohydrate does not exist isolately. It either combine with protein to form glycoproteins, or it may either combine with a lipid to form glycolipid, or it another layer which is considered to be a glycocalyx layer. As I said glycocalyx previously, so glycocalyx helps in tight fixation of neighboring cell. That is, it adheres the neighboring cell tightly together. Now let us discuss about the functions of cell membrane. In order to remember this function, you have to remember the mnemonic SLAP-ME. S stands for selective permeability, A stands for absorptive function, P stands for protective function, M stands for maintenance of shape and size, E stands for excretory function. Now let us discuss about the second important component, which is nothing but the cytoplasm. This cytoplasm, it is made up of 80% of cystosol, which is nothing but the liquid component, whereas a 20% is being made up of protein, carbohydrate and lipid. Now let us discuss about the organelle that is present in the cytoplasm. Among that, the first and foremost one is nothing but the endoplasmic reticulum. I hope so you know the structure of endoplasmic reticulum, which is more or like this. Okay, wow. So endoplasmic reticulum is considered to be a tubular structure. And so if this is endoplasmic reticulum, we have space here. This is considered to be a lumen. And this lumen of endoplasmic reticulum is being filled by the endoplasmic matrix. And remember, this endoplasmic reticulum forms the link between nuclear membrane and cell membrane. That is, endoplasmic reticulum connects the nuclear membrane with the cell membrane. Now let us discuss about the types of endoplasmic reticulum. So there are two types. First one is considered to be rough endoplasmic reticulum and second is nothing but the smooth endoplasmic reticulum. The endoplasmic reticulum is considered to be rough because here we have the attachment of ribosomes. So attachment of ribosomes make the group of endoplasmic reticulum rough and hence it is considered to be a rough endoplasmic reticulum. In smooth endoplasmic reticulum, we don't have any such ribosome, so hence it is smooth in nature. Now let us discuss about the function of rough endoplasmic reticulum. As I said previously, we have ribosomes over here. The very important functions of ribosomes is nothing but the synthesis of protein. And thus, the very important function of rough endoplasmic reticulum is nothing but the synthesis of protein. And second is nothing but degradation of worn out organelle, which is being performed by your rough endoplasmic reticulum. As we all know, so when a cell reaches a specific time, at that time what happens? That cell will degrade. And as a result, new cells develop in order to replace that old cell or degraded cell, right? Same happens even in the organelle also. So when an organelle has been destructed either by self or it is being damaged by any external events, that organelle is being degraded by your rough endoplasmic reticulum and your lysosomes. Okay. So now let us discuss about the function of smooth endoplasmic reticulum. So we have four important functions over here. So first one is nothing but synthesis of non-protein substance. Hence, rough endoplasmic reticulum involved in the synthesis of protein, and this involved in the synthesis of non-protein substance. First function. And you have to remember 3C over here. First C stands for cellular metabolism. So we have certain enzymes present over a smooth endoplasmic reticulum and that take part in the cellular metabolism. And second one is nothing but calcium. So smooth endoplasmic reticulum will act as a storage site of calcium. And third important function is nothing but catabolism and detoxification will also occur in the smooth endoplasmic reticulum. Now let us discuss about the Golgi apparatus, which is considered to be the neighbor of endoplasmic reticulum. That is, it lies very close to the endoplasmic reticulum. Now let us discuss about the Golgi apparatus, which is named after the discoverer Camillo Golgi. And this Golgi apparatus consists of 5 to 8 flattened membranous sac called cisternae. I have drawn only one cisternae here, and in the Madrid there will be 5 to 8 cisternae combined together to form Golgi apparatus. And this cisternae has two phases, namely one phase is considered to be a cis and second one is considered to be trans. Cis phase towards endoplasmic reticulum, whereas trans phase towards cell membrane. The phase which faces towards the endoplasmic reticulum is considered to be the cis. The one which faces towards the cell membrane is considered to be a trans. What is the function basically? As I said previously, this Golgi apparatus is considered to be a neighbor of endoplasmic reticulum. That is, it lie closely to the endoplasmic reticulum. So what happened here is, I said previously, what is the function of smooth and rough endoplasmic reticulum? That is synthesis of protein and non-protein substance, right? And those substance is being processed, later packed, and then it is delivered to the respective places by your Golgi apparatus. That is, the substance which is being synthesized by the endoplasmic reticulum is being processed, packed, and later delivery to the respective places. That is, either within the cell or outside the cell by your Golgi apparatus, and hence the Golgi apparatus is called as the post office of the cell as well as the shipping department of the cell. Now let us discuss about the rhizosomes. So rhizosomes will be more or less like this, right? So which is nothing but a vesicular organelle which contain hydrolytic enzymes within them. And this enzymes is being synthesized by your endoplasmic reticulum. As I said previously, the substance which is being produced or synthesis in the endoplasmic reticulum is processed, packed, and delivery to the respective places by your Golgi apparatus, right? So hence these rhizosomes are formed from the Golgi apparatus. Okay, and among all the organelle, rhizosomes has the thickest covering membrane. What is the types of rhizosomes? Generally, we have primary and secondary. So the hydrolytic enzymes which is present in the primary rhizosomes are inactive, whereas the hydrolytic enzymes which is present in the secondary rhizosome is considered to be an active one. The primary, when it fuses either with the phagosomes. I hope so you know what is phagocytosis, which is nothing but cell eating. So this primary lysosome, when it fuses with the phagosome, are endosome. So what is endosomes? As I said previously, phagosome is nothing but a vacuole or vesicle which is being formed by the process of phagocytosis, whereas endosome is nothing but the same vesicle or vacuole which is being formed by the process of pinocytosis. Okay, va? So this primary will fuse either with the phagosome or endosome to form secondary lysosome. And this lysosome is also known as suicidal back of the cell. And the important enzymes that is present in the rhizosome is nothing but plant. Protease, lipase, amylase, and nuclease. Now let us discuss about the functions of rhizosome in detail. Now let us discuss about the functions of rhizosome, which is nothing but the four important one. First one is the degradation of macromolecule. Second one is the degradation of worn out organelle. Third is nothing but removal of excess secretory product in the cell. And fourth is nothing but the secretory function. That is, when the secretory granule combined with the rhizosome, later that rhizosome is referred to as a secretory rhizosome. For example, this type of rhizosomes is present in melanocytes, in mast cell, and also in cytotoxic T cells and natural killer cell. Now let us discuss about the peroxisome. So peroxisome is similar to the lysosomes. That is, I said rhizosomes are the one which is formed from the Golgi apparatus, whereas the peroxisome are the one which is directly formed from the endoplasmic reticulum. And this peroxisome contains some oxidative enzymes such as catalase, urate oxidase, and deamino oxidase. And what is the function of peroxisome? Is nothing but, in order to remember the function, try to remember the mnemonic BAD FD. In that, B stands for beta oxidation. Second, D stands for detoxification. So it mainly detoxify hydrogen peroxide, and this peroxisome is present more in the liver as it is an organ of detoxification. And also this peroxisome accelerate gluconeogenesis, that is formation of glucose from fat. And also it degrades purine to uric acid. And also peroxisome helps in the formation of myelin and bile acid. Now let us discuss about the centrosome and centriole. Very easy. You don't need to learn more in detail about this. Centrosome, as the name suggests, it is an organelle which is present at the center. And centriole is nothing but there is two cylindrical structure. That cylindrical structure joined together to form centrosome, and those two cylindrical structure is referred to as the centrioles. And the movement of chromosomes during cell division occurs because of the presence of centrosomes.